So delighted to welcome Pauline from University of Poitiers as our first speaker, whose talk is off on VOC-based probes for cancer diagnosis and prognosis. She's a multidisciplinary researcher at the Institute of Chemistry of Poitiers. Her research group main objective is to deliver chemistry toolboxes for biology, and Pauline investigates chemical and biochemical strategies combining diverse omics approaches with the aim to highlight and monitor relevant biomarkers involved either in the origin or the basic processes of any living system. A recent development in research has been the identification of VOC-based probes targeting solid cancers with potential applications in diagnosis and prognosis. And Pauline is also a member of the Scientific Advisory Board here at Alstom. So Pauline, thanks for joining us, and we look forward to your presentation.
So I will present you a research program that we have launched 5 years ago in our research team, and which corresponds to the development of VOC-based probes for cancer diagnosis and prognosis. So humans are constituted of billions of cells in which thousands of chemical reactions occur simultaneously. With this chemical reaction, there are a lot of metabolites which are produced, and among these metabolites there is the volatile organic compounds. These small molecules are called volatile organic compounds because they are volatile when they are in the gaseous phase. And so when they are produced by the cells, these VOCs enter the bloodstream and then they are released in the air around the body. And this is these VOCs which are responsible for your odor, and this is because you are releasing a different mix of VOCs compared to your friends, to your children, to your neighbor, that you have a different odor from them. The study of these volatile organic compounds, which are produced and released by the living systems, is called the volatolomics. This science has been used in order to explore the biological processes which occur in real time in a body. The volatolomics has been used for different applications, among which there is the rescue and emergency application. Everybody has already seen in a film a dog who is smelling the odor of somebody in order to seek for this person, because the person has released behind him a mix of volatile organic compounds that the dog is able to smell. This is also used for disease detection. Indeed, cancer, viral infection, bacterial infections, diabetes, and asthma can be detected by a mix of volatile organic compounds which are released by the person who has the disease. For cancer, around 2,000 of VOCs have been potentially identified as candidate markers for cancer diagnosis. Among these 2,000 of VOCs, just a few ones are real markers of cancer. Indeed, there is 9 volatile markers for breast cancer, 19 for lung cancer, 3 for liver cancer, and 4 for prostate cancer. So the question is, why is there so many VOCs at the beginning of the study of the literature and then finally just a few ones? This is because there is a high inter-individual variability for the production of VOCs. Indeed, in function of your age, your gender, your sport habits, your diet habits, you will produce and release different mix of VOCs. And then, for example, it can be that somebody has no cancer and he releases during his basic metabolism a marker of cancer. And there is also another problem, which is the inter-laboratory disparity for samples preparation and analysis. And this disparity can lead to opposite results. Also, we do not know the specific metabolic pathways that generate these VOCs. And so, if we don't know the origin of the marker, this is sometimes difficult to identify the volatile as a marker. So for the moment, for the diagnosis, for the use of clinic of volatile compounds, just only combinations of multiple biomarkers can be used because they have a real predictive value. So in this context, we imagine that we can use an exogenous VOC to detect the cancer, and this exogenous VOC will be released artificially by the tumor in response to a tumor-specific stimuli. we have called this concept induced volatolomics. Here you have the representation. So you can see that there are some caterpillars that go to an injury, which is in the belly of the woman. And so when the caterpillar is in the injury, this caterpillar, which corresponds to a probe, will be transferred to a volatile organic compound, which is represented by the butterfly, and the butterfly will go in the bloodstream, and then it will be exhaled in the breath of the woman. In order to develop this concept, we need an enzymatic target, or a tumor target, and the one that was chosen is the beta-glucuronidase. So this enzyme is a good candidate because it is present in the tumor microenvironment, so in the extracellular space, when there are some cancer cells. In normal cells, this enzyme is present inside the lysosomes of the normal cells. So we can make the difference between the beta-glucuronidase present in the environment of the tumor cells and with the one that we can find inside the normal cell. This enzyme is present in the tumor microenvironment, probably because of tumor necrosis, and so it leads to the release in the extracellular space of the enzymes which are present inside the cell when there is a plasma membrane rupture. And it can be also present thanks to the immune cells that can release the enzyme in the inflammatory site. So here is the general concept. We first have developed a probe which can be substrate of the target that we have, which is to say the beta-glucuronidase. And so we have synthesis, the ethyl glucuronide in the liver. The ethanol will be transformed into ethyl glucuronide. And so when this metabolite is a natural metabolite, and when this metabolite meets the beta-glucuronidase, it will be hydrolyzed into glucuronic acid and ethanol. So here we get the idea to use an isotope of ethanol. So we have used the ethanol labeled with 5 deuteriums atom. So when we transpose the concept to an individual, so here, if you inject the probe, ethyl glucuronide, to an healthy man, this probe will pass in the bloodstream and then it will be directly eliminated in the urine. If you inject this probe to a man with a cancer, the probe will pass in the bloodstream and then it will meet the tumor. In the tumor microenvironment, it will meet the beta-glucuronidase, which will hydrolyze the probe into ethanol. Ethanol will pass in the bloodstream and then it will be released in the breath. So you can make the difference between a healthy man and a cancer man with the quantity of D5 ethanol that we will find in the breath of the pupil. So we have tested the concept on mice. Here we have selected the optimal dose to inject to the mice, and so this dose was 50 micrograms per kilogram. This is a very low concentration of probe to be injected to an animal. And so then we have injected this probe and we have measured the quantity of D5 ethanol that the healthy mice has released along the time. And so as you can see, along the whole experiment, there is no release of D5 ethanol in the breath of the animals. We have performed the same experimentation with tumor-bearing mice with a KB tumor, that is to say a human mouth carcinoma tumor. We have injected the probe to these mice and then we have checked the quantity of D5 ethanol that they have breathed out. Here you can see that there is an increased concentration of D5 ethanol in the breath of the animals when we have injected the probe, and this concentration is positively correlated with the tumor volume because the quantity of D5 ethanol increased as the tumor growth also increased. We wanted to check if this concept can work on other tumors, and so we have taken here tumor-bearing mice with a human mammary tumor or with a pulmonary tumor. And you can see that when we have injected the probes to these tumor-bearing mice release in their breath the D5 ethanol, while the healthy mice did not release the D5 ethanol in their breath. So with these VOC-based probes, the ethylglucuronide, we are able to detect different solid tumors in animals. Then we have tested the concept on transgenic mice, which are developing spontaneously a mammary tumor. And you can see there that we were quite disappointed because we did not have a difference in D5-ethanol concentration in the breath of both groups of animals. So we thought that there was no beta-glucuronidase in the tumor tissue of the transgenic mice. To verify this hypothesis, we have performed an immunostaining assay. Here you can see that there is no fluorescence on the tissue of the tumor of the transgenic mice, whereas we have seen the fluorescence on human mouth carcinoma tissue. So there is beta-glucuronidase in the KB tumor, whereas there is no in the transgenic mice tumor. Then we wanted to develop also an ANDI protocol using our probe to detect the enzyme on tissue. And so this protocol was really simple because we have taken the tumor, put it in an Eppendorf, we have added the probe, and we have monitored the quantity of D5-ethanol that can be released in the headspace above the tumor. And then we have noted that we have a release of D5-ethanol above the tumor tissue of human mouth carcinoma, whereas we have no release of D5-ethanol above the transgenic mice mammary tumor. So there is no beta-glucuronidase in this tissue. So then, as I was saying, there is a positive correlation between the quantity of D5-ethanol exhaled in the breath of the animals compared the tumor volume. So we wanted to see if we can use this probe to predict the efficacy of a chemotherapy. So to perform this experiment, we have used 4 groups of mice. One group was healthy mice. The other one was also constituted of healthy mice that were treated with the chemotherapy. A third group was tumor-bearing mice. And finally, a group of also tumor-bearing mice which were treated with chemotherapy. The chemotherapy that we have used here was a glucuronide prodrug, which targeted also the beta-glucuronidase. We have monitored during 26 days the weight, the tumor volume, and the D5-ethanol released in the breath of all the groups of animals. Here are the results. So you can see that the tumor-bearing mice treated with chemotherapy exhaled in average the same amount of D5-ethanol compared to the groups of healthy mice. So chemotherapy is working because there is no release of D5-ethanol in their breath. And compared to the tumor-bearing mice which were not treated with the chemotherapy, these mice were exhaling a large amount of D5-ethanol in their breath because their tumor grew a lot. Then, when we focused on tumor-bearing mice treated with chemotherapy, we had this evolution of tumor volume. Here you can see that there is an increase of the tumor volume during the first days of the experiment. Then after the treatment of chemotherapy, you had a decrease of the tumor volume. Just a few days later, again, an increase of the tumor volume. And so this is why we have treated a second time with the chemotherapy in order to decrease again the tumor volume. When you look at the quantity of D5-ethanol released in the breath of the animals, you can see that there is a clear correlation between the quantity of D5-ethanol that have been released in the breath of the animals and the tumor volume of the animals. Indeed, when the tumor volume increases, there is an increase of D5-ethanol in the breath. When the tumor volume decreases, there is a decrease of D5-ethanol in the breath of the animals. So we can predict the efficacy of the chemotherapy by monitoring the quantity of D5-ethanol in the breath of the animals. Then we want, as the concept is working very well, we wanted to transpose this concept to the clinic. And so for that, we wanted to develop a very handy protocol on blood sample. Indeed, as I was saying the target, the beta-glucuronidase, is present in the environment of the tumor. And so if this enzyme is present in the extracellular space, it can diffuse in the bloodstream of the pupil. And so we wanted to see if we can detect the enzyme in the blood circulation of the patients who had the cancer. And so for that, we have developed a protocol using the probe, which can also in blood sample detect the enzymes, if there is the enzyme in the blood sample of the diseased patients. So we have optimized the experimental protocol in order to detect the enzyme in the blood sample of the patients. And we noticed that we have a method performance with a limit of detection 100 to 1000 lower than the fluorescent substrates, which are targeting the same enzymes. So this is a really good limit of detection of enzyme. Then we have worked with the Clinical Investigation Center of the University Hospital in Poitiers, and we have recruited different patients, a group of healthy patients, 11 patients with lung cancer, and 11 patients with breast cancer. Within the group of patients with breast cancer, we had the patients which had a tumor with a volume inferior to 5 millimeters. So, during the protocol, we have first added probe and monitor the quantity of D5 ethanol released above the sample with water. Here, this is the blank sample, and you can see that when we added the probe to water, there is no release or a very little release or little noise of release of D5 ethanol above the water. Here is the healthy groups. You can see that when we have added the probe to the plasma sample of the healthy pupil, there is no release of D5 ethanol or just a noise above the sample. So there is no beta-glucuronidase detected in the blood sample of these patients. Here you have the results for patients with lung cancer and breast cancer. You can see that when we have added the probe in the blood sample of the patients with lung and breast cancer, there was a release of D5 ethanol above the sample. So we can detect the enzyme in the blood sample of these patients. And so we can, with this protocol, we can discriminate healthy patients from lung and breast cancer patients. Then, to go further, we wanted to enhance the robustness of our concept by designing a cocktail of probes. These probes will target different enzymes that can be potential candidate markers of cancer, and so we are focused first on the glycosidase. And with this cocktail of probes, we have imagined that we can detect these different glycosidase in the blood sample of cancer patients compared to the healthy patients. This cocktail is for the moment constituted of different probes that will target different enzymes that are the beta-glucuronidase, the N-acetylglucosaminidase, the beta-galactosidase, and the alpha-glucosidase. These enzymes are all possible markers of cancers, and so we have developed a probe which is constituted of the glycoside substrate of each of the enzymes, which is linked with ethanol. And so in order to discriminate between the different ethanol, we have used different isotopes of ethanol labeled with 13 carbon or deuterium. And so first we had to develop an analytical protocol to discriminate between these different isotopes of ethanol. When it was performed, we have also used this cocktail to discriminate healthy and breast or lung cancer patients with a blood sample. Here you can see that when we have added the cocktail inside the blood samples of healthy patients and breast and lung cancer patients, we had a higher amount of isotopes of ethanol released above the samples of the cancer patients. And so with this cocktail, we were able to detect beta-glucuronidase, N-acetylglucosaminidase, and alpha-fucosidase in higher concentration, in higher activity in the plasma sample of the cancer patients. And interestingly, maybe the N-acetylglucosaminidase can be used as a marker to discriminate between breast and lung cancer patients because we have a higher response lung cancer patients. This is just earlier results, and so preliminary results, and so we have to confirm this tendency. Then we wanted to see if this cocktail can work in vivo, and so we have also injected the cocktail to healthy mice and tumor-bearing mice. This mice has also a human mouth carcinoma tumor, and you can see that after 50 days after the tumor implantation, we had a release in the breath of the animals of the 4 isotopes of ethanol. That is to say that we can discriminate these tumor-bearing mice from healthy tumor by the measuring of isotopes of ethanol that are tracers of each of these enzymes. So there is probably beta-glucuronidase, fucosidase, N-acetylglucosaminidase, and beta-galactosidase in the environment of all these tumors. So now we know that we can use a cocktail to diagnose and also to make the prognosis of cancers, and so we have to continue this study. And for that, I will need help of all these pupils, which are all implied in the development of the results and the project, and so I really thank them for their help in this study. Thank you for your attention.
So the first question is, you mentioned that compared to the fluorescent substrates, sensitivity limit of detection is much better. But if you're targeting an enzyme in systemic circulation, are there other approaches to do that? What's the advantage of using the volatile probe compared to other types of approaches?
So the sensitivity of the approach is really good. And so, for example, are you talking about the microbiome of the intestine? For example, if we want not to detect this type of cells, this is what you are mentioning now? Yeah.
No, it's more to do with the principle of, rather than putting a probe in the body and looking for the released volatile reporter, where you're talking about removing the sample, looking for a target enzyme in systemic circulation. So the question is, why a volatile reporter is better than other approaches that you might use?
I think that it's better. It is not really better, but we know that to inject some probes in the circulation, we need to complete some administration and administrative steps, and so using the blood sample is easier, and so this is just to facilitate the transfer of the concept. So this is why we are now working on blood sample. But I think that we should have the same results in vivo and ex vivo, and so this is just we just work on blood sample to facilitate the transfer of the concept in the hospital in France. Yeah.
That makes a lot of sense. In one of the talks on liquid biopsy yesterday, part of the challenge when you're taking the sample out, you have dilution, and if the enzyme is originating in the tumor microenvironment, you've got to hope it's been in the circulation and then it's coming out in the sample as well. So that makes a lot of sense. I suppose, obviously, from a regulatory standpoint, it's easier to administer a cocktail of probes or multiplex probes in vitro compared to in vivo. But you think that ultimately putting compounds in vivo is likely to yield better results? Is that what's your kind of prediction there?
I don't know if the in vivo use of these compounds will give some better results than ex vivo, but I know that they give comparative results, but I'm not really sure that you can be more sensitive with the in vivo compared to the ex vivo, because we are really sensitive with these probes, and so we are able to detect enzymes at a level of 10-18 mole. So 10-18, this is a very low quantity of enzymes that we are capable to detect. So I think that we should have some comparative results. And the advantage also of using these probes ex vivo is that we can use this on biopsy, on tumor tissue, and so it can be used to select the patients that can answer to a treatment also. So this is also interesting I think to use these both methods, these both approaches, in vivo and ex vivo, to have a wide, a deep cartography of the tumor.
Got it. Thank you. So obviously one of the big advantages of a unique compound like D5-ethanol is it's unique. So you shouldn't see it in the ambient. And that's one of the things that we've been talking about in some of the endogenous VOC sessions. So in one of your charts, you showed where the probe was administered to a blank, which was just a water blank. And there was D5-ethanol being produced there. I'm wondering what the mechanism of that is and whether or not that's suggestive that you would have a background of D5-ethanol present in healthies as well as disease.
In healthy mice, you are talking about in vivo mice there?
No, I think it was the plot where you included the small breast cancer. So the sample was collected and you put the probe into the blood sample. And before that, you had a water blank. I think it was a deionized water blank. And there was at time point 2, 3, 4, there was some D5-ethanol. So I'm wondering where that's coming from if the enzyme's not present.
So we have a kind of natural hydrolysis of the probe, because the problem of the glucuronide, this is not really a problem, but you can have some spontaneous hydrolysis of the probe. This spontaneous hydrolysis is due to the presence of the carboxyl groups on the probe, and so then you can have sometimes an hydrolysis of the probe, but this is a very low quantity of probes that can be hydrolyzed. And we have performed some experiments to reach the dose for which the probe is not hydrolyzed. And so the quantity that we have put in the sample, in the blood sample, is the minimal quantity that we can which will not generate a spontaneous hydrolysis of the probe. But this is normal because you have sugar, and so sugar can be hydrolyzed.
Got it. Thank you. Next question is from Robert. How sensitive do you expect this approach to be with early stage diagnosis, and with regards to cancer in the colon and the possible involvement of the microbiome? Are you considering strategies to target these microbial populations that might be present in larger numbers compared to cancerous cells?
So we can have we have thought about other probes that can be specific to the bacteria cells, and so these probes will not be the same as the one that we are using now for the cancer cells. And so by using these two groups of probes, we can make the difference between the microbiome and between the cancer cells.
Got it. Thank you. Next question here relates to the regulatory side of things. Do the probes require the same regulatory approval as a new drug would?
Yeah. So as I have answered this question, but this is quite easy to get the approval of these probes, because we just need safe compounds. That is to say, a natural sugar and a natural VOC, and so the combination of both compounds make the probes to be rather sure, also, but rather safe. And so this is easier to transfer the probes to an in vivo use.
Got it. Thanks. I think this question you covered in the presentation, but I'll just read it again. How large does the tumor have to be to get a measurable amount of D5-ethanol in breath? In other words, could the probe be able to detect early stage lung cancer?
Yeah. So I don't know for lung cancer, but as I was saying in the presentation, we have detected a tumor which was 2 millimeter. So this is a really early diagnosis tool, and so we can expect to have the same sensitivity on lung tumors too.
Brilliant, thank you. And 2 questions from Chad. Did you check for the levels of D5-ethanol in the liquid phase of the blood samples? And do you think you could do dynamic analysis to show enzyme kinetics in the blood samples?
Yeah, so we always have performed the kinetics of the enzyme. This is also why we know that this is the enzymes that have answered with the probes, because we have always performed the kinetics, the enzymatic kinetics with the probes, and so we are pretty sure to have a kinetic of enzymes on the sample, and so we are also able now to monitor the level of the enzymes in the blood samples with the probes. That is to say that we are able to observe the decrease of the enzymes after treatment in blood sample.
Got it. Thank you. And then the last question. So when you think about mechanisms to selectively target a cancer, so obviously therapeutics companies have been trying to do this for a long time, where they're trying to deliver a toxic payload. Do you think that there's things that we can learn from the therapeutics field rather than delivering a toxic payload? We're looking at a volatile reporter coming out. Do you mean that these kind of probes can be toxic if we inject these probes a long time? This is what you are meaning there? No, I mean where drug companies are trying to develop therapeutics for cancers, they're obviously trying to selectively target the tumor and kill the tumor. But do you think there's things that we can learn from those companies, the targeting mechanisms, but rather than trying to kill the tumor, obviously what we're trying to do in a diagnostic standpoint is signal that the tumor is present?
Yeah, I think that with this kind of probes also we can study the metabolisms of the tumor in response to the treatment, and we can also, I think, imagine to follow to monitor the dynamic and the aggressivity of the tumor with these kinds of probes. And so we are now thinking about a set of probes to study these metabolisms, yes, in response to a treatment or not. And we are also thinking about probes which can help us to understand the mesenchyme, the change to some stem cells.
That's question from Scott. In vivo, have you seen any metabolism of the D5 ethanol?
No. Yeah, never. Yeah.
Okay. From John, how long do you give the probe before sampling?
How long do you
do you probe before sampling? So I think it's the interval between administering the probe and then collecting the sample.
If we are talking about the time are you talking about the time that I have to keep the probes before using it?
No, I think the question is when you administer the probe, yeah, and then take the sample, what's that time interval?
That's just 1 hour. When we are injected the probes in vivo, we have to expect 1 hour before measure the quantity of D5 ethanol in the breath. And when we are using the probes ex vivo, we are performing kinetics, and so I think that we have to expect 4 hours after the injection of the probes to get the D5 ethanol signal.
Got it. Thanks. And the last question, I think, is anticipating the next talk as well from Yuri. Could we apply this approach for other diseases which aren't cancer?
Yeah. So we are now thinking about bacteria and viral infections like the COVID-19. And so, yes, we are working on other diseases.
Wonderful. Excellent. Well, thank you so much for that, Pauline. That was really fascinating talk, and thanks for answering the questions.